So Mendel used letters to denote the different particles in the hybrid. So let's just call the yellow particle capital Y and the green particle a lowercase y. The full genotype for the hybrid then, if this is in the F1, the full genotype for this hybrid would be big Y, little y. Now, it's very important to note that alleles of a locus, so the different versions of a given gene, always use the same base letter. So it wouldn't be appropriate here to use, say, a Y and a G for yellow and green. If we're going to talk about these two particles as alleles of the same gene, then we need to specify that they are the same gene and alleles of the same gene by using the same letter. If we use a different letter, that would be confusing. So when you're picking out your characters for assigning to alleles, make sure you use the same letter for different alleles of the same locus. Okay, let's work through how we explain the 1:2 to 1 ratio in the F2. So the F1 is just the cross between two heterozygotes. And we can ask the question, what is required to form an F2 individual with a big Y genotype? Now the first requirement is that we have to get a big Y from the pollen, and we also have to get a big Y allele from the ovum, the second requirement. And you'll notice that we say "and" in between those two. The next thing to say is, well, what are the probability that these events will happen? Well, it's a 50-50 shot either way if the pollen is going to have a big Y or a little y, and same for the ovum. So each of these events or requirements has a probability of 1/2. To determine the final probability, we need to apply the product rule. And the product rule is simply a rule that states that the probability of independent events occurring together or in a sequence is the product of the probability of the individual events. So we have these two requirements, and when we list them off, we have to say "and" in between them. We need to get a big Y from the pollen, and we need to get a big Y from the ovum. So the probability that these events will happen is 1/2 each, and to apply the product rule, we multiply them together. So this means that for the F2 progeny, with a genotype of big Y, we expect 1/4, so 1/2 times 1/2. Let's do one other example. The little y case is exactly the same as the one that we've just done. The one that's more tricky, on the other hand, is the heterozygote case, where we actually have the production of big Y, little y individuals in the F2, or the yellow hybrid seeds that Mendel recognized in his crosses. In this particular case, there's two different ways that we can make up this genotype. The one way is to get the Y from the pollen and get the big Y from the pollen and the small y from the ovum. But at this point, then we have to say "or," because there is another way that this could happen. We could also have a small y from the pollen and a big Y from the ovum. And when we're faced with that "or," that tells us that there's two distinct ways that we can form this particular genotype. So each of these different paths that could give us the thing that we're looking for, the big Y, little y individuals in the F2, each of these paths, mutually exclusive paths, have an individual probability of 1/4, calculated using the sum or the product rule as we've just talked about. But because these are two mutually exclusive ways, in order to get this genotype, either one way or the other, we need to apply what's known as the sum rule in this case. And the sum rule states that the probability of either of two mutually exclusive events occurring is the sum of their probabilities. What we'll do now is we'll explore a method known as a Punnett square to graphically show how these different probabilities can be determined. Okay, so let's show how we can use a Punnett square to represent what's going on in an F1 self cross. So an F1 self is simply crossing the heterozygous individual with itself. So this is the cross that we're looking at. And a Punnett square is just a very convenient way to represent the gametes and the offspring involved in the cross. What you do with a Punnett square is you take the gametes from one of the parents and put them down the one side, and then the gametes from the other parent across the top. The other thing that a Punnett square allows us to do is assign probabilities to each of these gametes. And as we mentioned before, they each have a probability of 1/2. Then a Punnett square is simply a way to show the frequencies and the different genotypes that result when these gametes combine. So in this case, we've got the big Y individuals, and that's just the big Y gametes coming from both parents and combining to give a big Y individual. Likewise here we have heterozygotes, another heterozygote here, and then the little y category. Determining probabilities is quite straightforward. It's simply just the product rule that we're applying. What does it take to meet a big Y individual? Well, you need a big Y gamete and another big Y gamete. And because we say "and" in between those, that indicates that we use the product rule. So that gives us a probability of 1/4 for this particular genotype. It's exactly the same logic down here. What does it take to be little y? Well, you need little y from one, little y from the other. So again, 1/2 times 1/2 equals 1/4. Now, what does it take to be heterozygous? Now here's the difference. There's two different ways for this to come about. Each of them has an individual probability of 1/4, but either way satisfies our expectation of having a heterozygous individual. So if we say, what does it take to be heterozygous? Well, it could be this way, or it could be that way. And that "or" indicates that we're going to use the sum rule. So we could list it as a set of requirements. We could say probability, 1/2 times 1/2 equals 1/4 for the one root. 1/2 times 1/2 equals 1/4 for the other root that we haven't. And then because of the sum rule, where we say we could have it this way, or the same, if it happened this way, it would give us the same result. That "or" indicates that we use the sum rule. So we add the two probabilities together. So taken together, what this shows is how we arrive at the 1:2:1 ratio that we expect for this type of cross. We have, in reality, what this is showing us is it's a 1/4 to 1/2, to 1/4 ratio, which is mathematically the same as a 1:2:1. Okay, now that we've seen Mendel's model as he has developed it thus far, let's just show one other experiment that he did as a test of the model that he had developed. And he chose this cross because it made a specific prediction of what he should see. This type of cross, where he took an F1 hybrid yellow and he crossed it to a pure breeding green, you can see that this is going to give us a different ratio than what we saw when Mendel crossed two hybrid yellows together. And Mendel did this cross because he expected to see this different ratio, and it would act as an independent confirmation of his ideas that the hybrid had two discrete particles. So again, we can use a Punnett square to easily represent what we would expect in this case, putting the gametes from the one parent across the top, the gametes from the other parent down the side. And in this case, what you'll see when you fill in the Punnett square is that we would expect half of the progeny to be yellow, and in this case we'll have the other half that are green in a 1:1 ratio. So Mendel actually did this cross, and when he did this cross it produced the 1:1 ratio that he was expecting, acting as a further confirmation about his ideas about what sort of particles were present in the hybrid. A test cross is something that you should be familiar with because it's often used in the types of genetics problems that we'll be solving in class. A test cross is a very easy way to see what is present in, say, an unknown genotype, because the homozygous recessive individual in the cross does not contribute to the phenotype. You'll notice that whether or not a progeny is green or yellow is solely determined by what allele is coming from, in this case, the hybrid, the unknown. The little y allele does not contribute to the genotype. It's strictly determined by what allele comes from the hybrid in this case. If we get the big Y, then it will be yellow. If we get the little y from the hybrid, then pairing up with the little y will give us green. So it gives us a nice way to test and see what genotype an unknown individual might have.